Today, we're going to talk about mitosis! Woo! Mitosis! Mitosis! Sorry, Parker. We're actually not going to look at mitosis today. We're going to look at something very similar, but also slightly different. And that is meiosis. Guys, we know that mitosis is how we made body cells. Well, meiosis is going to be the process for us to make sex cells, like eggs, sperm, things that we're going to call gametes. So if we take a look at this video today, we're going to go through the steps of meiosis cell cycle. We always start with interphase. Guys, no matter if you do mitosis or meiosis, interphase is going to be first. We're not going to see chromosomes. The cell is going to have loose DNA, and it's going to be when it does its job and just copies that DNA that's in there already. Then when we start meiosis, we're actually going to begin with prophase just like mitosis. However, we're going to call this prophase 1 because we're going to do everything twice. In prophase 1, the chromosomes are going to be visible. You saw that in that little image right before this. Chromosomes are going to condense and become visible. Those homologous chromosomes are going to pair up and get all close and cozy together. Right? One from the mom, one from the dad. And they're going to get so close and so cozy that they're going to have something called crossing over happen. So if you look at this little stop motion, okay, do you see how close they're all getting so cozy together? And the blue one that says from the dad and the red one that says from the mom, they're all buddy, cozy, snuggy, wuggy. And something's going to happen because they're so close together. We're going to call this process of these chromosomes exchanging pieces crossing over. So because of that closeness, they're going to swap pieces of chromosomes, which might seem weird, but it's a good thing because now neither chromosome is 100% mom or 100% dad. It's a little bit of both, which makes for more genetic diversity. The other thing you can see in prophase 1, just like in prophase of mitosis, is that the nucleus is fading away. Get rid of it. We don't want it. So remember, crossing over is something that happens during prophase 1. It is a positive thing because it's exchanging genetic material. It's making genetic variability increase. So we have more sperm and eggs that are different than the same. You can see the crossing over in these chromosomes. Each piece has been kind of swapped with the homologous pairs. Okay, so a little piece of the mom's chromosome is on the dad, swapped with the blue and red that you can see there. Okay, now that we're done with prophase 1, we're going to move on to metaphase 1. And just like mitosis, metaphase is going to have the chromosomes lined up in the middle. Those spindle fibers are what moved them there. And the only difference, let's go back real quick, between metaphase 1 of meiosis and metaphase of mitosis, dang it, is that look how they're lined up. They're still paired together. The homologous pairs are still cuddly wuddly, cozy, snuggly, right next to each other. And that's going to help us when we go to anaphase and pull apart our chromosomes. We won't pull apart the sister chromatids. So in metaphase 1, look at the difference. The chromosomes are lined up next to each other down the middle. So now that we move to anaphase 1, because it's still the same order, prophase, metaphase, anaphase, each whole chromosome is going to move to an opposite end of the cell. So look at the entire chromosome is being moved to the left, or the entire chromosome is being moved to the right. We're not splitting sister chromatids in anaphase 1. Those spindles move the whole chromosome, and now we're ready for telophase. In telophase 1, once again, telophase, 2 new nuclei are going to form around those chromosomes. And you can see that new nuclear membrane reforming here. We'll perform cytokinesis and split our 2 cells. All right. And we're going to restart basically. So the problem is we're not done. Those chromosomes are still duplicated. They're still X's. They still have sister chromatids attached, and that doesn't work for us. So we basically need to restart. All right. We're not going to copy our DNA again, but just make sure we have enough organelles for each new cell that we're going to split one more time. And we are going to redo meiosis, those same 4 steps, to end up with 4 cells. So here we are again. The DNA is going to condense into chromosomes. The nucleus is going to go away one more time. So now we're at prophase 2, and this is going to look really similar to prophase of mitosis. You have chromosomes. They're not in homologous pairs anymore. We split the homologous pairs in meiosis 1. Crossing over already happened. You can still see the pieces that are different. Okay. And after the nuclear membrane goes away, the chromosomes are going to move to the middle, which is metaphase. So in metaphase 2, just like in mitosis, the chromosomes line up along the middle of the cell, okay? And that one looks just like mitosis. Now in anaphase, we're going to pull apart our sister chromatids. Anaphase 2 looks like anaphase of mitosis. Guys, look at each sister chromatid being pulled apart to opposite ends of the cell. So this one looks more like mitosis because in anaphase 1, the homologous chromosomes are being pulled apart. So now that each chromosome is being pulled, we're going to move to telophase. 2 new nuclei are going to form in each cell. So if you look at this, remember, we started with 2 cells at the beginning of meiosis 2 because we already did meiosis 1. So now each of those cells is going to split, and we're going to end up with 4 cells at the end here. So a new nuclear membrane is going to form around each of those chromosomes. The chromosomes are going to decondense, go back to spaghetti. Cytokinesis will split apart all 4 of those cells. So at the end of meiosis 2, we're going to have 4 daughter cells. Remember, daughter doesn't mean girls, just means new baby cells. Remember that these 4 cells are sex cells, gametes, eggs, sperm, and they are genetically different. Each of these is unique, not only because of that crossing over, but because one half of the dad's chromosome got pulled into this, and then maybe one of the moms, and then one of the dads, and then another one of the dads, and one of the moms, and each one is unique and different. They are not clones of each other. That's how we get all of our children to be different, even though you guys are related to your siblings. You're not identical twins. So at the end of meiosis, we have 4 haploid, thanks Carolina and Catherine. We have 4 haploid daughter cells. Remember, haploid means they have half the normal number of chromosomes.